able to see in focus. I can't.
So, in the back,
So sometimes these guys squirt.
your retina, which is where the image is projected, was this
Sounded gross.
So a little bit of vitreous humor is coming out,
at the opposite.
and the easiest way to dissect these is to kind of circumscribe around the cornea.
Here, which projects back into the
There's a soft space between the sclera, which is hard and firm. When you hold this thing, even now, I've opened it up, and the sclera is a cup and it's retaining its shape. This gets really quite loose. It's fixed, but the cornea is this loose, really loose, flaccid material.
bright colors.
So the liquids that are coming out, what's coming out is vitreous humor?
It might be formaldehyde, but if it were a fresh, dead eye at body temperature, vitreous humor is liquid. Aqueous humor is liquid. We've fixed this, and we're at room temperature, so the the aqueous humor takes on this jelly-like consistency. But in life, it's liquid. It helps maintain intraocular pressure, to keep the shape of the cornea, and to keep the retina planted in the back of the eye, and to keep the what's going on? Is that you? And to keep the distance between the lens and the film at the right focal length. This is a camera, and we have two lenses. Let's go through it. So if we travel the path of light, light's going to come, strike the cornea, and be refracted, bent. I won't go into which angles because it's different for each species, but let's say they get bent towards the middle. So it's a converging lens. It passes then through a liquid substance called aqueous humor, which would be between the cornea and the lens. Now we got a couple structures in here. This first structure is the iris, circular muscle, smooth muscle in a circular pattern that's able to relax and constrict. And in the space of the iris is called the pupil. And when we constrict the iris, we constrict the pupil, the size of the pupil. And when we relax the iris, we dilate the pupil. So the pupil is not a structure, the pupil is a space. All of us have different eye colors. We got brown, bluish, greenish. Can't tell, you're too far away. Bluish, greenish. I got blue, but all of our pupils are black due to the absence of light. We'll get into that in a second. So we've passed through the pupil, and we hit our next lens, called the lens. This is circular. The cornea is really flat. This is a much more powerful lens than the cornea is. We're able to accommodate the lens, meaning change the lens's shape. So we can see things far away when the lens is flat, or see things close up when this lens is circular in structure. A circular lens is a powerful lens. The muscle that controls that is called the ciliary body, or the ciliary muscle, which kind of got cut away in my dissection. But we see a little bit of it here. It's in black. Oh, I can see. It's hard to see. I'm going to try not to touch anything, but it needs to be in the light. You'll see fine fibers. It's almost like a shadow radiating away from right in here. Do you see fine fibers radiating away from the lens? Anybody?
Yeah.
Those are ciliary processes, and they are very fine. And they will extend to the ciliary muscle, which is going to surround the lens, and based whether it's contracted or relaxed will change the shape of the lens. Now I'm going to peel this lens out. The vitreous humor is going to come with it, and more than likely the retina is attached to the vitreous humor, and the retina is going to pop out, but we'll put it back in place. So there's lens, and it's kind of encapsulated in vitreous humor, and this yellowish substance, which I'm going to try to put back in place, is your retina. I'm doing a really bad job. There, kind of spreading out. That's not too bad. So retina, this yellowish tissue in the back, this is an extension of brain, consisting of a dozen different nerve types, rods and cones being probably the most familiar. And the rods and cones are going to synapse on bipolar cells, horizontal cells, amacrine cells, and ultimately ganglion cells, synapse, 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 all interneurons. And then the axons of the ganglion cells all merge together at the optic disc. Which direction is our optic disc? There we go. So I've taken the retina and just kind of swept it up into one side of the eye cup. But it's still connected to the eye cup. And it's actually passing through the eye cup right here, the optic disc structure or space through which all of these ganglion cell axons form the optic nerve, and the optic nerve is going to project out the back of this. And off we go to the chiasma.
So it's not really like defined, it's like where it goes through.
The disc, yeah, if we were to put it back together again, you can imagine, I'm doing a bad job, but if we were to put it all back together again, where the optic nerve is emerging out of the eye, that's the optic disc, and then we have a blind spot there, a lack of vision, where all these ganglions are coming together. There's no photoreceptors there. So we have a blind spot, and you can find it. Right? There's a little trick to find it. A piece of paper, and you hold it here, and you put a black dot on it, and you move the paper, you stare at something in the corner, you move the paper until the black dot disappears. And you can find where your blind spot is. Maybe we'll try that in lecture. So we have photons passing through all these lenses, a certain focal length to get onto the retina, where it's going to form an image by striking the rod and cone photoreceptors, isomerizing the visual pigments found within, and they send a signal ultimately out to the optic nerve. And then the photons keep on going through the retina, and they strike this tissue back here. And we have kind of two colors here. I'll kind of turn this guy like that. We have black pigmentation, and that's pigment epithelium. And we see it here, and we also see it over here. And then we have this pretty mother of pearl color, and that's the tapetum lucidum, right? The tapetum lucidum serves to allow the retina to get struck by a photon of light a second time. So it's a mirror. Photons go in, pass through the retina, fire rods and cones, hit the tapetum lucidum, and scatter back out, firing rods and cones again on the second time out. It doesn't result in a clear, acute, defined image. It scatters them, but you see a photon of light. You might not see a jaguar chasing you in focus, but you can see the jaguar chasing you. So it's good for nocturnal vision. We don't have that. We've lost that. Our whole back eye cup is made of pigment epithelium, and that is the eye. Good? You got that all on videotape?
The extrinsic muscle was what's surrounding
The extrinsic muscles are out on the outside. They help the eye move. Seen a little bit better on the model of the eye.